substrates, you can usually find a slightly different version of the nucleotide which will bind but then not do the job, and then that will act as an inhibitor. The second type of inhibitor that we can use is a short piece of nucleic acid that binds to that internal guide sequence and competes with the recognition of the exon by the intron. And again, that works very well in the test tube, but I'm not sure that anything like that normally goes on in cells. So in the cell, I don't know if there are competitors that are of significant problems for the system. Yes?
Yeah, I was wondering if the ribozymes like mimic the protein enzymes in that the rate of reaction is affected by pH and heat. Have you all done any tests for that?
pH and
Heat.
and heat. Yes. So the question is, does an RNA catalyst change its activity as you change pH or temperature in a way that you might expect from studies of protein catalysts? And the answer is yes, and those dependencies are often rather revealing about how the reaction works. For example, we find that as you go to more and more from acidic conditions, more and more towards basic conditions, the rate of the reaction speeds up a lot, which suggests that you've got to pull a proton off of some entity in the molecule in order for the reaction to occur. And that's easier to do in a more alkaline solution. If you have a more acidic solution with a lot of hydrogen ions around, you're putting hydrogen ions onto the molecule, and that makes the reaction slow down. We think that the key proton that has to be removed is from the guanosine itself, that OH that the H has to be removed from the OH in order for the guanosine to act as an effective pair of scissors to cleave the RNA chain. And the temperature dependence, as in many enzyme reactions, as you raise the temperature, the reaction goes faster and faster. It's very common for chemical reactions. And then if you raise the temperature enough, it stops completely. And why do you think that is? Why at high temperature might the reaction not work at all? Go ahead. In the blue shirt.
It denatures it.
Right, it denatures it. So that, as I've been trying to stress, the folded structure is critical for catalysis. At a high enough temperature, entropy takes over, the chain unfolds, and now it can't do anything. So you get this interesting temperature dependence as you go to temperatures that are above body temperature. The reaction towards what you'd find in a hot spring in Yellowstone or something, the temperature, the reaction goes faster and faster, and then you get to sort of temperatures closer to where water boils, and the reaction quits entirely because the catalyst no longer retains its shape. So that was a good question. Yes?
When you extend the leg or the arms of the ribosome, how do you know which base pairs to use?
So the question was, when we extend the arms of the ribosome so that we can visualize it in the electron microscope, how do we know what base pairs to use? And we didn't spend a lot of time worrying about that. We just made a stretch of A's, U's, G's, and C's, and then much further on, we made a sequence that was complementary, so that there would always be a match going across the helix. You might imagine that you'd have to worry a lot about it, because if you didn't choose exactly the right sequence, it might fold in some other way, in some branched way, rather than as a straight rod. And so you do use computer programs to check out your sequence and see if they predict that it'll fold as a simple rod or possibly in some alternative way. And we chose a sequence. The computer program said it would fold correctly, and according to the electron microscope, it looks like most of the time they fold correctly, maybe not 100%. In the back.
Does RNA behave like a protein enzyme in that it has to be it's unchanged after use, or does it have to be reintroduced into the system over a period of time?
So the question is, does an RNA enzyme act like a protein enzyme in that it will keep going and going and going, sort of like the ever-ready battery, or does it have to be reintroduced to the system after a certain amount of time? And in principle, it's limitless in the number of reactions that it can catalyze. We've tested it, for example, after the catalyst has been working overnight. We've added fresh substrate RNA, and it continues to cleave the added substrate at the same rate that a fresh batch of the catalyst taken right out of the refrigerator. So it doesn't appear to have lost any of its activity overnight. But I think the real answer is that for either a protein catalyst or an RNA catalyst, there will always be some competing pathways. For example, denaturation or unfolding of the catalyst itself, and so you don't expect it to. That's why I said in principle it would. You would never have to restore it. It would always keep working. In practice, nothing is ever 100% dependable, and in this case, there would be some loss of activity over time. But I would say it's similar to a typical protein catalyst in that regard. Way in the back.
Was magnesium the cofactor that helped the rate the most out of the metal ions that you tried?
Question was magnesium ion the metal ion cofactor that was most effective of the metal ions we tried? And the answer is that for the normal RNA substrate, not the altered one with the sulfur, either magnesium or manganese have very similar activity. When we substituted the sulfur, we found that zinc worked quite well, and zinc is known to be a thiophilic or sulfur-loving ion, and so that fit in with the model that I presented and provided some further evidence for it. Other metal ions that are common in cells, like calcium ion, are not able to substitute. And so there is a moderate amount of specificity for particular metal ions, and it does not have the ability to use just any metal ion that would be in the cell, but it seems to be choosing in the natural form either magnesium or manganese. Last question, anyone? Yes?
Have any of the RNA catalyzed reactions been known to speed up during cell division or other cell processes?
You mean as if they would be regulated?
Right.
And so the question is, have any of the RNA catalysts been shown to speed up during cell division or in a particular cellular state? And the answer is that there's been a lot of talk about such possibilities, but I think the truth is there's no clear example for regulation of a ribozyme activity as a function of the cell cycle or some other environmental condition. Well, thank you very much for your excellent questions.
I want to thank Tom for his lucid presentation on his work on the structure and function relationships of catalytic sites of ribozymes. His next talk will focus on his work on RNA, whose target is the ends of chromosomes, which hold the secrets not only for the aging of the cell, but paradoxically for its immortality as well.